Good afternoon, everyone. I am Jaya from Abhilashi University from the Department of MLT. I would like to express my sincere gratitude to Abhilashi University and NDLA Club for giving me opportunity to deliver this lecture. Today, I am going to speak on a very important topic, that is inflammation, that affect all of us in our daily lives. Inflammation, a natural response to injury and infection. Inflammation is the immediate response to injury and infection that aims to eliminate or limit the spread of injurious agent as well as damaged tissue. Let us begin. Contents, introduction, causes and signs of inflammation, types of inflammation, different mediators of inflammation, lastly, conclusion. Introduction. Inflammation is the local response of mammalian tissues to injury. When any part of the body is injured, tissues at that specific areas reacts only. Inflammation is necessary for the survival of the host. In the absence of inflammation, body would not be able to kill or eliminate the injurious agent. So, inflammation is one of the innate defense mechanism of the body. Term inflammation is derived from Latin word which means Latin word inflammare, which means to burn. However, burning is the one of the feature of inflammation. Inflammation is known by the suffix itis. Itis indicates inflammation and is added to the name of the inflamed organ. For example, inflammation of liver, hepatitis, inflammation of skin, dermatitis. These are different images showing inflammation at work. Knee inflammation, inflamed face, inflamed foot. Inflammation is general protective response by the body to different etiological agents that may be infectious or non-infectious. Inflammation involves host cells, blood vessels, proteins, and different mediators that eliminates the injurious agent, limits the spread of injurious agent, followed by removal of dead cells and tissues. This is the image of a damaged tissue. Damaged tissue release histamine, a mediator, to increase the flow of blood at the affected area. Histamine causes capillaries to leak, releasing phagocytes and clotting factors into the wound. Phagocytes engulf bacteria, dead cells, and cellular debris. Then platelets move out of the capillaries to seal the wounded area. Causes of inflammation. Causes of inflammation include physical agents, chemical agents, biological agents, immunological agents, and inert materials. Physical agents include mechanical injuries, change in temperature and pressure, injuries through radiation exposure. Chemical injuries agents include organic and inorganic toxins, biological agents, bacteria, viruses, fungi, parasites, immunological agents like cell-mediated and antigen-antibody reactions, inert material, foreign bodies, dirt and sutures, etc. Inert materials indirectly associated with the inflammation. Signs of inflammation. Roman medical writer Celsus in the 1st century described four famous cardinal signs of inflammation that are seen at the site of injury or infection. These are rubor, redness, tumor, swelling, calor, heat, dolor, pain. To these fifth sign, punctulatia, loss of function, was later added by Galen, a pupil of Hippocrates. This is the image showing different signs of inflammation. When finger is injured, skin over inflamed area appear red. Inflamed finger feels warm to touch. Finger appear swollen. Person feels pain when touching or moving the finger. Scab, a hard dried blood clot formed at the affected area. This is the image of damaged tissue of inflammation. When any harmful object or pathogen enter into the body, it produces different signs. Redness. Redness is caused by dilation of arterioles. Heat or warmth is due to the increased chemical activity and increased blood flow to that area. Swelling caused by accumulation of fluid and damaged tissue, blood cells at the affected area. Dolor or pain, direct injury of nerve fiber or blood collected outside the blood vessels cause pressure, lead to pain on the nerve endings. Increased pain, swelling, and decreased activity cause loss of function. Depending upon the host capacity and duration of response, inflammation is classified into two types. First, acute inflammation. Second, chronic inflammation. Diagram showing two phases, acute inflammation and chronic inflammation. Acute inflammation is of shorter duration, include burns, cut, allergic reactions, infection, chemical irritants, frostbite, exposure to cold temperature. Chronic inflammation is of longer duration. When acute inflammation becomes persistent, chronic inflammation is seen in chronic diseases. These are autism, rheumatoid arthritis, autoimmune diseases, depression, neurological diseases, Alzheimer, cancer, and cardiovascular diseases. Acute inflammation. Acute inflammation is of shorter duration, persists of few minutes to few days, represents early body reactions and resolve quickly. Most often, it is a healthy response. Its main features are accumulation of fluid and plasma at the affected site, activation of platelets within the vessel, polymorphonuclear neutrophils as inflammatory cells. Major events of acute inflammation include vascular event and cellular event. Vascular event is further divided into two categories: hemodynamic changes Altered vascular permeability. Cellular events include exudation of leukocytes, phagocytosis. Vascular events. Vascular events are early and essential changes that occur in blood vessels at the site of injury or infection during inflammation. These alterations or changes include first, hemodynamic changes, second, altered vascular permeability. Hemodynamic changes are the earliest feature of inflammatory responses, results from changes in vascular flow and diameter of these vessels. Sequences of hemodynamic changes is as follows. First, vasoconstriction, narrowing of blood vessels, vasodilation, dilation of blood vessels, local hydrostatic pressure. 4. Stasis. 5th, leukocyte margination. After injury, these after injury, there will be an immediate vascular responses, including transient vasoconstriction of arterioles. In mild flow of injury, blood flow re-established within 2 to 3 seconds. But in severe case of injury, it lasts for 5 minutes. Vasoconstriction is followed by vasodilation. Vasodilation occurs after half an hour of injury. It involves mainly arterioles. Vasodilation results in increase in blood volume, which results in redness and warmth at the site of inflammation. Local hydrostatic pressure, increased blood volume in turn elevates hydrostatic pressure. Pressure exerted by the fluid against wall of the blood vessel. This pressure results in transudation of fluid into extracellular space, which is responsible for swelling at the affected area. Fourth, slowing or stasis. Slow movement of blood increase permeability of microvasculature that results in increased concentration of red blood cells, leading to increased viscosity of blood at the site of inflammation. Fifth, leukocyte margination. Leukocytes stick to the vascular endothelium and then move and migrate through gaps between endothelial cells in the extracellular space. Next is the altered vascular permeability. In and around the inflamed tissue, there is an accumulation of edema fluid in the interstitial compartments, which comes from blood plasma by its escape through endothelial walls. Initially, escape of fluid is due to vasodilation, which is later followed by an increase in vascular permeability, allowing plasma proteins, immune cells, and other components of blood to leak out in the surrounding tissue. Diagram showing vascular permeability and mechanism of vascular permeability. Mechanism of vascular permeability consists of following points. First, contraction of endothelial cells. Contraction of endothelial cells develops temporary gaps between them due to their contraction. This response begins immediately after injury. Contraction of endothelial cells affect venules, capillaries, and cause damage. Third, direct injury to endothelial cells. Direct injury to endothelial cells cause cell necrosis and appearance of physical gaps at the site of detached endothelial cells. This change affects venules, capillaries, and arterioles. 4. Endothelial injury by polymorphonuclear neutrophils. Adherence of leukocytes to the endothelium at the site of inflammation causes activation of leukocyte. Activation of leukocyte release different proteolytic enzymes which cause endothelial injury and increased vascular leakiness. It is a late response. Neovascularization, newly formed capillaries under the influence of growth factor release, process of repair become excessively leaky, and later on endothelial cells mature and develop junction. Cellular events of acute inflammation. Cellular phase of acute inflammation include the recruitment of leukocytes at the site of infection, their activation to recognize the microbes, and eventually phagocytose and clear off them. Cellular phase of inflammation consists of two processes. First, exudation of leukocytes. Second, phagocytosis. Exudation of leukocytes is the escape of leukocytes from bloodstream into the site of tissue injury through the wall of blood vessel. It is the most important feature of inflammatory response. In acute inflammation, polymorphonuclear neutrophils form the first line of defense, followed later by phagocytes. Steps involve margination, rolling, adhesion, transmigration, chemotaxis. Margination. Normally, blood cells flow centrally in the blood vessels. In case of inflammation, blood flows slowly and leukocytes move toward vessel wall. White blood cells, particularly neutrophils, bind to endothelium and marginate due to change in blood flow. Rolling, peripherally marginated neutrophils slowly roll over the endothelial wall. Rolling along the endothelium facilitated by lectin and corresponding ligands on the endothelial wall and on the surface of neutrophils. Adhesion. Neutrophils firmly bind to the endothelium through interaction between integrin on the surface of the leukocyte and adhesion molecule such as ICAM first on the endothelial cells. Transmigration. Leukocytes squeeze through the endothelial junction and migrate through endothelium to reach at the affected site. Chemotaxis. After exiting the vessels, leukocytes migrate toward the site of injury or infection by chemotactic signals released by damaged cells, bacteria, or other inflammatory mediators. Chemotaxis is essential for the recruitment of immune cells at the site of inflammation. At the site of infection, when there is an infiltrated leukocytes, now the process of clearing of microorganism starts. That is phagocytosis. It is the process of engulfment and degradation of solid particulate material by a cell. It involves three steps. recognition and attachment, engulfment, killing and degradation. Phagocytes recognize and bind to the microorganism through surface receptors on macrophages. After attachment, they are bound to the phagocytes and engulfed by phagocyte by the formation of cytoplasmic pseudopods around the particle. This is called phagosome. Phagosome fuses with one or more lysosomes of the cell to form bigger vacuole phagolysosome. After this stage, microorganism is disposed of, which is the major function of phagocytes. Microorganisms are killed by antibacterial substances, are later degraded by hydrolytic enzymes. Second type of inflammation, chronic inflammation. Chronic inflammation is a prolonged and persistent inflammatory response which lasts for weeks, months to years. In this process, tissue destruction, inflammation, and repair occurs at the same time. It involves macrophages, lymphocytes, and plasma cells. Chemical mediators of inflammation. Chemical mediators are substances released from cell membrane from the cells of plasma, damaged tissues during inflammation. These are broadly classified into two groups, cell-derived mediators, plasma-derived mediators. Cell-derived mediators. Cell-derived mediators are released either from their storage in the cell granules or are synthesized in the cell. Various Various cell-derived mediators are vasoactive amines, arachidonic acid metabolites, lysosomal components, platelet activating factors, cytokines, nitric oxide, and oxygen metabolites. Vasoactive amines. Two important vasoactive amines are histamine and serotonin. These play important role in early inflammatory responses by increasing vascular permeability and vasodilation. Arachidonic acid metabolites. These are released from cell membrane by phospholipases. These are essential fatty acid constituents of cell membrane. They produce several inflammatory mediators, include prostaglandins, leukotrienes. Next, lysosomal components. Inflammatory cells like neutrophils and monocytes contain lysosomal granules, which produce a variety of mediators of inflammation. Platelet activating factor. It is released from basophils, mast cells, leukocytes, and platelets. Platelet activating factor is a key mediator in inflammation, promoting various vascular changes, leukocyte recruitment, and activation of immune responses. Cytokines. Cytokines are the small proteins acts as a chemical messenger within the immune system. They play an important role in regulating inflammation as well as directing immune responses. Nitric oxide and oxygen metabolites. Nitric oxide and oxygen, reactive oxygen species are released from activated neutrophils and macrophages, and both act as a potent mediator of inflammation by increasing vascular permeability and inducing relaxation of blood vessels. Plasma-derived mediators include products derived from activation and interaction of four interlinked systems. These are following: kinase system, clotting system, fibrinolytic system, complement system. Factor 12 of clotting system play a key role in the interaction of four systems. Kinase system. This is the flow diagram of kinase system. Factor 12 activates factor 12A that generates bradykinin. Bradykinin acts in early stage of inflammation, and its effects include increasing vascular permeability, contraction of smooth muscles, and dilation of blood vessels. Clotting system. Factor 12A initiate cascade of clotting system by the formation of fibrinogen. Fibrinogen is acted upon by thrombin to form fibrin and fibrinopeptides. Fibrinopeptides acts by increasing vascular permeability, chemotaxis for leukocytes, and anticoagulant activity. Next is the fibrinolytic system. This system is activated by plasminogen activator. It acts as on plasminogen present as a component of plasma proteins to form plasmin. Plasmin break fibrin into fibrinopeptides or fibrin split products. Fibrin split products increase vascular permeability and are chemotactic to leukocytes. Complement system. Complement is a group of serum proteins mainly synthesized in the liver and widely distributed among body fluids. Complement system comes into effect only when it is activated. Critical step in activation is the proteolysis of C3. It can occur by three pathways: classical pathway, alternate pathway, lectin pathway. This is the diagram showing complement system. These three pathways, alternate pathway, classical pathway, lectin pathway, activate C3, a complement protein, cleaved into C3A and C3B by C3 convertase. C3B bind to the surface of the pathogen. So, pathogen is easily recognizable by phagocytes and removed by phagocytosis. C3B is converted to C5B by a series of reactions which form membrane attack complex. This membrane attack complex cause lysis of cell by forming pores in the cell. Conclusion: inflammation is the body's alarm and repair system. Without it, infection would go unchecked, wound would never heal, and injured organs remain as a permanent decaying lesion. So, understanding inflammation, its signs, stages, and mediators help us to regulate inflammation for developing effective treatments and maintaining overall health. Lastly, inflammation is the body's fire, essential for protection but dangerous if it burns out of control. That's all. Thank you.